synaptic transmission. So the thing you keep in your mind that it means transfer of action potential. Okay, transfer of action potential from one neurons to another neuron. Now how? We have to understand the process, how it's happening. So for that we have to depict one diagram. Okay, diagrams make things easy to understand. Fine, but I need a space. Let's have a bit. See. This is one neuron's axon part. You can say the synaptic end part. Fine. And I'm just making another different color. Okay. This is two neurons. Now, this space, okay, you know the measurement of this space called synaptic cleft, and 15 to 20 nanometer space referred as a synaptic space. Now, what things this presynaptic neurons is holding? Yeah, they are holding first the mitochondria, a membrane bound organelle. Fine, mitochondria. Okay, and they have the synaptic vesicles. They have the synaptic vesicles. Now, what is happening? The thing is which is happening is action potential from any muscle, any gland, okay, or from you can say the centers, okay, the centers which is present in your brain, okay, they are commanding. Once they are receiving, you maybe you remember the process which I soon tell you. First, central nervous system should receive the information, fine, then they process the information, then they respond the information. Yeah, if you are not understanding, just understood that in the previous lecture, okay, we have the playlist in neuroanatomy and neurophysiology. You can go and watch that. For functioning better, okay, for functioning you can say accurately about the central nervous system, then central nervous system should work on these three principles. First, it should receive the information, okay, and here the central nervous system is receiving the information from the gland. Yeah, it is receiving the information from the gland. Then on the way one neurons met with the another neurons by this synaptic, you can say by the forming the synapse, okay, and then there are neurons, okay, and by this they goes to the central nervous system. Once then again central nervous system is process the information, then send again via these synapse, they come to this gland. Understood? No problem. Now we have to understood action potential. Fine, action potential they came. Okay, action potential is come to this axon. Fine, if it is myelinated neurons, then it comes through the process of saltatory conduction. Yeah, jumping saltatory conduction. Okay, because there is a myelination. Yeah, so action potential jump from, you can say, the one node of Ranvier to another node of Ranvier to the further. Fine. Now, this action potential, they open one channel. Okay, they open one channel. Remember that there are channels in our cell membrane. Okay, there are channels in our cell membrane, which is, you can say, working. Which is a main, you can say, the part, okay, which is the main part of present in any cell for their proper functionality, for the maintenance of their proper functionality, there is always influx, okay. Efflux means sometimes there will be a inward movements of the ion, and sometimes from the, you can say, the environment, they have to, you can say, throw out the ions in order to maintain the equilibrium, okay, in order to make the harmonious environment. Sometimes cell have to thrown out some of the charges, I can say, yeah, and sometimes it have to receive the charges in order to maintain the equilibrium. So for this, for serving this purpose, there are channels, okay. We have the ionic channels, okay, and we also have the receptors, ionic channels, and we have the receptors. So one of the ionic channels is here involved, is a calcium channel. Now once the action potential they reaches, okay, they will stimulate this calcium channel, they will open. Initially they were closed, but once they will receive the action potential, fine, these channels become functional, they will open, and from the extracellular environment, they receive the calciums, and calciums goes inside, okay. Once calcium, the first, okay, side by side, you just also write it down, the process is what is happening, fine. So first thing what we are seeing, the action potential arrival, isn't it? Fine, the action potential arrived. Then what is happening? By the action of this action potential, there is the opening of calcium channel. Opening of calcium channel. Okay, no problem. Now once the calciums comes in this environment, fine, calciums, these are white, I am showing dots as a calcium. Once the calciums comes in the environment, what is happening? They will stimulate one of the protein, okay, that is called as the clamidulin, okay, calmodulin, okay, it's called as calcium binding protein, okay. And now this calcium, they will bind with the clamidin protein, okay. So there will be a binding of clamidulin. Now calcium won't escape, it is a binded. Now what is happening? The negative, you can say, environment which is present initially, once they are receiving the calcium, now their negativity is overcome, okay, and it become the hyperpolarized, fine. Initially 65 millivolt, how much? Minus 65 millivolt current, you can say, is present inside, and inside is referred as a, you can say, the deep, you can say, polarized state. They are in a polarized state. But once the calcium, there will be an influx of calcium, okay, inward movement of calcium, and you know that calcium is a positively charged molecules, so there will be a increase in the charge, and this become minus 45, yeah, minus 45 millivolt. And now this membrane become excited, fine. So there is a depolarization of presynaptic membrane, okay, depolarization of presynaptic membrane. Depolarization, remember we are talking about the presynaptic membrane. Now let's take example that in these synaptic vesicles, the neurotransmitter which are present is acetylcholine. Now you know already the process. The acetate is synthesized in the mitochondria. Cholines comes from the GIT. They combine and form the acetylcholine, and they are seated. Fine, no problem. Now there are proteins. Okay, there are proteins which is present in this environment. One of the protein is called as synaptobrevin. Synaptobrevin. brevine, and one of is syntaxine. Now, the syntaxine is present in the membrane, okay, and calcium, they comes and they fit it over here in these parts, okay, their end they will fit it over here. They stimulate the membrane. Synaptobrevine, they will bind with synaptic vesicles, yeah, and once they bind, then there is a fusion of these vesicles, because already the membrane is activated by your syntaxine protein, understood, and this both protein, remember, they will only activate on the presence of calcium. Are you following? Synaptobrevine, synaptotaxine, the major protein, okay, they will only activate only the presence of calcium. Am I clear? No problem. Now come. The synaptotaxins, they are present in the membrane, okay, and they have the, you can say, the ligand part, fine, where the calcium love to fit over here, okay, calcium love to fit here. Once calcium find, membrane become stimulated simultaneously, okay, or we can say the meanwhile this synaptobrevine, they will you can say attach with these vesicles, okay, and there will be a docking. So that's amazing term. I was also previously not known this term. That's called the docking process. Docking means like attachment, fine, and this is the docking site. This site is a docking site, yeah. Now this synaptic vesicles, they will fuse, okay, this is your synaptic vesicles, they will fuse on the synaptic part, and there will be a, yeah, exocytosis of content which is holded by these synaptic vesicles, yeah, content which is holded, that's your acetylcholine. Now acetylcholines comes into the, and remember I am just showing you one synaptic vesicles where they, but for one excitatory purpose they are in a, you can say, larger amount as well. Many synaptic vesicles once upon a time they will fuse with this membrane, okay, and this business is ongoing, but I am just showing you one. Now the synaptic content or acetylcholine comes in this environment. Now what is the next process? The next process is we have to talk about this membrane. What is the special present in this postsynaptic membrane, okay, which is waiting for the arrival of acetylcholine to you can say start their function, start their business. What things is present? The thing which is present is receptor, okay, and since it is also a membrane, so it's also holding some of the, yeah, it's also holding some of the channels. The one channel is sodium channel, fine, one is the sodium channel and one is the chlorine channel. These channels are also present in this membrane as well, fine, but this action potential, the stimulus is adequate, okay, the threshold is adequate, it stimulates the calcium channel. Now these receptors for the acetylcholine receptor, we are talking about the acetylcholine, then mark this receptor as a acetylcholine receptors, fine. Now this acetylcholine, they will bind over here, they will bind over here, fine, and once they will bind, it means there is a transmission of action potential, okay, from the synaptic cleft to here. Then what is happening? There will be a changes in the, you can say, the charge of this molecule. Initially these were also sitting, okay, this is also sitting, okay, they are basically in the inhibitory state, but once they receive the charge, the minus 90 millivolt current which is present here. We are talking about the, you can say, the skeletal muscles. That's why this action potential is minus 90 millivolt, the resting membrane potential. We are referring it as a resting membrane potential. When the membrane is not doing any work, then if we measure their charge, then their charge is minus 90 millivolt. Fine. Once they receive, okay, their charge is going to minus 45 millivolt. But how? Just because there is a formation of neurotransmitter receptor complex, okay, and it will stimulate again further the sodium channel, and there will be an influx of sodium, and you know the sodium is positively charged, then it create minus 40 millivolt current. Fine. Now it becomes in excited state. The message action potential it traverses again. But the thing is, sometimes what is happening, there is here also there is a presence of one enzyme. Okay, there is a presence of, you can say, the enzyme, and these enzymes previously we discussed already. This is called as acetylcholine esterases. Fine. The acetylcholine esterase function is, okay, I'm in a plenty of amount, the vesicles, which is, you can say, the docking, fusing, okay, in a larger number, but that much, you can say, the neurotransmitter is not needed. That much neurotransmitter is not needed. Then what is about the rest neurotransmitter? Because this is the duty of the, you can say, the presynaptic neuron itself to synthesize. It means it have to do something in order to save their neurotransmitters. So this acetylcholine esterases, they will break the acetylcholine into esterate and choline, okay, and that is reabsorbed. That is reabsorbed by the presynaptic membrane. That's fine. Now, what is happening in the case of myasthenia gravis, that's autoimmune diseases? There is a formation of you can say, the antibody against these receptor. Fine. So if this receptor is occupied by these antibodies, then acetylcholines are not able to bind. Are you following? Okay, they are, if it is bind, okay, this myasthenia gravis, they produce this remember, this is the autoimmune diseases. Fine. And they are producing the antibody, okay. These antibody love to fit over these acetylcholine receptors. They mimic. Fine. They mimic as this neurotransmitter, acetylcholine receptor, and they will bind over here. But these neurons, they are secreting their content, but these neurons, they won't show any activity. Fine, because this receptor is blocked by this antibody. And this enzyme, acetylcholine esterases, they are in a functional. Fine. They are thinking, okay, this much you can say amount is came in my synaptic cleft environment, and I think they are thinking, okay, the postsynaptic membrane they already use, and rest of the, you can say, the synaptic neurotransmitters I have to you can say break. So they are working, but they don't know that, my God, these receptors which is occupied by antibody, it's not by the acetylcholine. So one of the pharmacological management is making this acetylcholine esterases non-functional. Okay, so once they are non-functional, then the neurotransmitter which hold here, and again there are immunosuppressive drugs which suppress this antibody. Fine. And the person have bit relaxation. So this is about the myasthenia gravis. Understood? So once again, what is happening? Write it down here. Depolarization of presynaptic membrane. Then there will be a docking, Fine. Then after the docking, there is exocytosis. No problem. Now this exocytosis lead to the binding formation of, normally not the disease condition, forming the receptor neurotransmitter complex. Yeah, neurotransmitter receptor complex. Fine. And there will be a, you can say, the depolarization of the postsynaptic membrane. Depolarization of postsynaptic membrane, and there will be a further action potential is transported. Understood? No problem. And what you are observing, is it inhibitory kind of synapse or excitatory? Yeah, very good. It is a excitatory. Excitatory synapse. Excitatory synapse. No problem. Fine. Understood. So once again, just revise it. What is happening here? Action potential coming. Okay. They in turn open the calcium channel. Fine. There will be an increase in the charge from minus 60 millivolt. It goes to minus 45 millivolt. There is a binding of calcium with the clamidin protein. Calcium is trapped. Okay. By this calcium, there is a two protein which is present called as a synaptobrevin and synaptotraxin. Okay. It will stimulated on the presence of calcium. Okay. Synaptobrevin will bind with the synaptic vesicles. Synaptotraxin is, you can say, present in the presynaptic membrane where the calcium binded. Then there will be a docking, exocytosis of the neurotransmitter which bind with the postsynaptic receptor. There will be a formation of neurotransmitter receptor complex. Okay. By this there is activation of sodium channel in the postsynaptic membrane. The charge will increase from the resting membrane potential minus 90 millivolt. It goes to the minus 45 millivolt. Okay. And there will be a further transfer of the action potential. Now the problem is that sometimes what is happening, the myasthenia gravis patient, they secreting some of the antibody which will, you can say, love to fit over this acetylcholine receptor. Fine. And there will be a blockage on this receptor. And there is an enzyme, okay, which is helping to absorbing the rest of the, you can say, residual neurotransmitter. This is acetylcholine. They are breaking. We are referred as the acetylcholine esterases. Fine. So in the case of myasthenia gravis also they are not non-functional. Only the receptors become non-functional. Fine. So one of the treatment in the myasthenia gravis is that to making somehow by the pharmacological agent this acetylcholine esterases non-functional, so that the neurotransmitters can hold here. Okay, can hold here, and whenever they got chance they go and bind because they are their natural place. These are not natural, but the acetylcholine, this is their natural place. Fine. Understood? No problem. And because they are involved in the excitation, we are referring this kind of synapse as a excitatory synapse. Fine. We are referring these kind of synapse as a excitatory synapse. No problem. The only thing which you just correlate here, okay, now about the inhibitory function. What is happening in the inhibitory function? There will be an influx of, in the postsynaptic membrane, there will be influx of chloride ion. Okay, and chloride ion, you know, it is a negative. So it become, you can say, inhibited. Okay, it is only in inhibited stage because there will be only opening of chlorine. Okay, chlorine channels. Understood? That is about the, you can say, the inhibitory synapse. Understood? No problem. That is about the inhibitory synapse. There will be opening of the this chlorine channel. Understood? No problem. Now, once you understood the synaptic transmission